Yeah, okay, I'd like to thank Ben for a nice introduction and the kind of invitation. Yeah, so I'd like to talk about my journey of the nanomechanics. Yeah. So actually, many years ago, when I was a PhD student, I have been mainly work on like hardness, grip, and flux toughness, and interface failure of thin coatings, and develop different models to determine flux toughness based on different type of fractures, such as picture frame crack, and also many other different fractures. Later, I think biological materials is more exciting. So although many of you may not agree, you may think high temperature materials may be more exciting. So then I decided to work on nanobiomechanics of engineered tissues and also native tissues, such as, for example, bone cartilage. And later on, I guess because I'm small, so I decided to move down to smaller particles like cells and bacteria. So today I would mainly give two typical examples about the nanoindentation of the cells and bacteria. Yeah, so basically for here, I would choose some examples about the cancer cells. So why we are interested in the cancer cells? Yeah, I guess everyone knows. Like for example, for the breast cancer cells. So basically, 1 in 8 female would suffer from breast cancer at some point. So there's always some argument about if cancer cell is stiffer or softer, than its healthy counterpart. Actually, some people believe it needs to be softer so that it can migrate, into the human body. But there are also some evidence, which suggests opposite. Then the other thing is also quite important for us to know the cell ECM or cell biomaterials adhesion, because actually this would also affect the cell migration, how the cell grow, and also the, for example, like cancer cell emission as well. So regarding for the indentation of the very small particles like cells or bacteria, so in that case, we need to use the AFM. But all those fundamental contact mechanics would still apply. So when we consider indentation, so most of us basically is using this. Is using this solid cantilever. But actually, I would also like to highlight. So nowadays, we can also use this hollow cantilever. So basically, that means, so for this cantilever, basically it's like microfluidics. So we can apply the negative pressure so that we can detach the cell from the substrate. So now the key question is, how can we determine the viscoelastic properties and cell materials adhesion simultaneously just based on a single test? And also how these properties can be altered by the underlying substrate. So you can think about for biomedical applications, or you can also think about for the disease progressions. Because when there's a disease, actually the extracellular matrix mechanical properties would also change. So basically for here, to choose some representative biomaterials, for here we use PDMS. The reason we choose it, basically for sure, this is biocompatible materials, have wide applications for the as the biomaterials, and we can also easily alter the stiffness by changing the cross-link density. So in this case here, so we choose the stiffness range to be between 0.1 and 2 megapascal. So here I just try to briefly explain how this hollow cantilever works. So basically, usually when people use this hollow cantilever, they just use it to detach the cells. But actually when we do this measurement, when we try to detach the cells, so we need to hold the cantilever usually for a few seconds. So for those few seconds, basically clearly we can see the stress relaxation. And then we think, okay, we can just take advantage of this stress relaxation, which would allow us to determine its viscoelastic properties. On the other hand, we also take advantage that the cantilever is also transparent. So that means that we will be able to observe the contact area between the cell and the cantilever. So basically for here, I just show you this simplified equation. But actually, in reality, we also did a correction of this equation because we also need to consider the complicated cell morphology as well. So in that case, yeah, basically after this equation, actually there is also some empirical parameters as well that I didn't put it here. So now clearly we can say very strong stress relaxation that's correlated to the viscosity of the cells. And when we do the detachment test, so basically we can also say, basically all these events is associated with some adhesion containing structure detachment. So for here, just in case you may not be familiar with the biology, so basically for the NCF7 cells, so that's the breast cancer cells. Then for the NCF10A, so that's the healthy adipocyte cells. So now when we look at the equilibrium modulus and the instantaneous modulus, so just make it easier to understand. For the equilibrium modulus, so you can think about when you stretch the material is extremely slow, so basically you get the equilibrium modulus. For instantaneous modulus, you can think about is step loading, so that's the instantaneous modulus you will get. So based on these results, so basically you can say for the cancerous cells, actually it shows a stronger materials dependent, the structure dependent behavior compared to its healthy counterpart. On the other hand, we can also say when it is on the stiffer substrate, so basically the cancerous cells appears to be a bit stiffer for both equilibrium modulus and also instantaneous modulus. When the substrate becomes a bit softer, then there is no significant difference. So now we also try to check, how about the results we can find in literature. Actually, in literature, people always culture those cells on rigid petri dish, and they use those spherical indentation to do the large deformation. So in that case, what they found is the healthy cell is stiffest compared to the cancerous cells. But for here, I would also like to highlight is in those indentation tests, so basically they use spherical indentation, they do large deformation, and they just simply use the Hertz model. They didn't consider any viscosity effect and also the geometry effect. Let me also try to find what else we can find in literature. So then later we found people also use a different technique, that's called the scanning ion conductive microscope, to measure the localized properties. So when they take the average the localized properties, so they also found that basically the relative stiffness between the healthy and the cancerous cells, actually it is materials dependent. Let me also look at the adhesion. So regarding the adhesion between the cell and the subject, so for here we can say for the healthy cells, they always have a stronger adhesion force and also adhesive work. So adhesive work is to do the integration of the detachment curve. So basically, this is also correlated with the migration test. So for here, we do the single particle tracking. So we track the individual cell movement, then we can calculate its average speed. So from here, you can also say basically for the cancerous cells, it migrates much faster. So this is correlated to its lower cell materials adhesion. Just briefly summarize this part. So basically for here, so we notice for the cancerous cells, actually it has a stronger substrate dependence compared to its healthy component. And also if we consider for the given type of cells, so for example, consider either cancerous cells or the healthy cells, basically there is a bit of correlation between the equilibrium modulus and also its adhesion to the materials. So basically, this is not a surprise because when usually for the given type of cells, when you have a stronger adhesion, usually the stress fiber is also better aligned. So usually that would also lead to the higher modulus as well. But we wouldn't do this kind of cross comparison between different type of cells. Also for the cancer cells, it has a weaker adhesion, so which is also correlated to much higher migration. So for all this technique here is for the cells, but actually we think this can also be applied to engineered materials as well. Okay, so now I will talk about bacteria. So basically, for bacteria, it's quite important. So if we think about, for the life at extreme environment, so basically, there are some bacteria, they can survive in high temperature or very low pH, for example, like bacteria in your stomach. And also some bacteria, they can also survive in high pressure, and even some relatively strong radiation as well. So for all these could be related to the mechanics as well. On the other hand, when we also consider antimicrobial resistance, actually according to the World Health Organization, antimicrobial resistance can kill more people than cancer and diabetes combined. So basically, when we think about bacteria, so you can also image it as engineering materials, except it has quite complicated structures. So for example, this is a typical example for gram-negative bacteria. So it has a multiple layer of its cell envelope, and actually inside the bacteria, there is also high internal pressure called the turgor. It can be up to 4 atmosphere pressure. So in literature, so most people, they just do the indentation test and either use Hertz model or Snyder model to get the stiffness. But for us, what we are thinking about is how can we determine the cell wall stiffness and the turgor simultaneously, because both properties are very important for bacteria to survive. And also how the turgor may affect the apparent cell modulus. So for here, I just give you one example about how we indenting subdermis. So basically, subdermis is also responsible to a lot of infections, such as skin infections. So in principle, so when you try to get the mechanical properties, you can either use pyramid probe or spherical probe, depending on what kind of probe you use. So we will use completely different mathematical modeling. Certainly, not just Hertz model or the Snyder model. Those model wouldn't work for this complicated case. Okay, so if we just use the pyramid indenter, actually for here, in order to determine the cell wall stiffness and also turgor, so we also did the finite element simulations as well. So now if we compare the FE simulations and also some analytical solutions, for example, called like the resonance model. For resonance model, it was originally developed for engineering materials, but the key point is it assumes it is point loading. But when you indenting bacteria, which is so tiny, it's around 0.5 micro to 1 micrometer, so it's not point loading. So that's why we have to modify that equation. And actually, we also notice basically if we increase the turgor, basically the apparent Young's modulus of the bacteria also increase as well. So basically, once we modified that model, so now we can say we can predict the force displacement curve when we indenting the bacteria. Then what we found is when we put the bacteria in different osmolality conditions, so basically we can say the cell wall stiffness actually it will decrease when we increase the osmolality. Actually this paper has already been published 3 years ago in nanoscale. And then we do the FE simulations with iterative adjustment. Then we can also determine the turgor. So again, the turgor also decreases with the increase in the osmolality. So basically, for given type of bacteria at given chemical conditions, So when we consider the bacteria as a population, so basically for the individual bacteria, its cell wall stiffness also correlated to the turgor as well. Then actually when we do the indentation of the bacteria, we can also use the spherical indenter as well. So for here, in order for us to use the analytical solutions here, so we need to choose the spherical probe, which needs to be large enough. So in this case, like 8 or 10 times of the bacterial radius. So in that case, we can adopt these analytical equations. Actually, before we use this here, we also did FE simulations here to validate it. And after we did FE simulation, actually, we also did the experimental work on the engineering materials to validate the equation as well. So as you can see, so basically there's a good agreement. Then we applied it to the bacteria. So the beauty is, it doesn't matter if we use the pyramid probe or if we use a spherical probe. So for those bacteria in different chemical conditions, it gives us very consistent results for both cell wall stiffness and turgor. Okay, yeah, so for here, just like to say, we already demonstrated when we combine modeling and nanoindentation, so we'll be able to simultaneously determine the cell wall stiffness and the turgor. And actually, when you change the ions for the culture medium, so that would also affect the cell wall stiffness and the turgor. So as long as we use the appropriate equations here, so no matter if we use a pyramid or spherical probes, so we can get the consistent results for cell wall stiffness and the turgor. Actually, we also believe regarding the potential applications, so we can use this to study the effect of antimicrobial drugs on the bacteria. And actually for the modeling framework we developed here, that can also be used for the engineering materials, such as capsulate. Okay, so in the end, I would like to acknowledge all my group members and also collaborators. Yeah. Thank you for attention.